Thank you and hello everyone. The talk being presented today will be by my colleague and co-director at NCNED, Professor Sonya Marshall-Gradisnik, and myself. Unsurprisingly, the topic of our talk is on transient receptor potential or TRP ion channels in ME-CFS. And we're going to describe a picture of how we developed our understanding of TRPs in ME-CFS. When we started our work at NCNED, our mission was very clear that patients would be our priority and that we would have three aims. These are to discover the pathomechanism of ME-CFS, to develop a valid and reliable test, and identify suitable treatments, including potential drug repurposing. And as it turns out, the Nobel Prize for 2021 for Physiology or Medicine was awarded to Drs. Julius and Patapushin. And in a slide courtesy of the Nobel Prize Committee, you can see the original research that was done by these researchers on one of the TRP channels here. And on the right, you can see a number of channels associated with heat, and these include the TRPMs, TRPM3, TRPM2, and TRPM8. The Nobel Prize Committee noted that the knowledge that was acquired from these discoveries is being used to develop treatments for a wide range of disease conditions. First, we'll look at the discovery of the pathomechanism of ME-CFS. Now, TRPs, as we know, are ion channels. They're located on every cell in the body, and they convert external threats or stimuli into biological activity. And these threats are well described, and these can be from toxic chemicals, vapours, fumes, infections, trauma, even surgery, infections and irritants, and even temperature. And the way these TRP channels work is to transport calcium, magnesium, or sodium into the cell. And this calcium passes into the cell where it can then affect a number of biological functions. And these TRPs are located throughout the body, and all tissues and organs have some dependency on proper functioning of TRP channels for them to work, for those organs to work correctly. And as we've seen with the distribution of TRPs, in particular the brain, endocrine tissue, the gastrointestinal tract, urinary system, and muscle tissue are critically dependent on these functions. These then correlate with the types of symptoms that we see developing in people with ME-CFS. Initially, we discovered that there were abnormalities in TRP genes known as SNPs in patients with ME-CFS. So the question that we had was, is there a change in expression of TRP ion channels on the cell surface of cells from people with ME-CFS, and what was the impact on calcium entry and storage? On the left there, we can see that in detecting TRPM3 on the surface of the cell, particularly in this case on bright CD56 natural killer cells, there is a significant difference between healthy controls on the left and the cells from ME-CFS patients in the middle. There's also a change in the cytoplasmic storage of calcium. And when those tests were undertaken using various stimulants, we could see that there's a statistical difference in the calcium that's transferred into the cell. Now, calcium is critical for the development of NK cell function. So as bright CD56 cells on the left, in the most proliferative phase, move to a more cytotoxic phenotype, calcium is primarily responsible for that transition. And on the right, we can see calcium coming into the cell by the TRPM receptor, TRPM3 receptor, and that can either be promoted or inhibited by various drugs. We also looked at the gastrointestinal tract. And in this slide, on the top left-hand side, you can see a negative control of a healthy control person. On the right-hand side, using the same staining mechanism, the findings in a person with ME-CFS. However, when we look at TRPM3 specifically, when we look at a healthy control, we can see quite prolific distribution of TRPM3, but when we look at a patient with ME-CFS, there is a far reduced expression of TRPM3 in that tissue. And when we look at brain function, when challenges are made to concentration by patients versus healthy controls, you can see on the bottom line, when various challenges are provided, there's limited stimulation of the brain that's recorded, whereas in ME-CFS patients, they have to recruit far more brain regions to achieve a similar result. So cognitive tasks require more brain regions to be recruited in ME-CFS versus healthy controls. And that signalling is further reduced in ME-CFS. And one of the most critical regions that's identified is the RAS, or the reticular activating system, which has a number of very important functions in terms of attention, consciousness, memory, and sleep quality. So I'll ask Professor Marshall-Gradisnik to take on from here.
Thank you, Don, and welcome those people that are attending in person and all those people who are in attendance virtually. So in terms of our discoveries in relation to ion channels and ME-CFS, we were the first to confirm significant changes in expression of TRP ion channels on the cell surface, as well as calcium entry and storage in cells, in particular natural killer cells. We've confirmed this again in gastrointestinal tract cells, as Professor Stone's outlined. And for that reason, we endeavoured to achieve that objective to see if what we were seeing on a natural killer cell was similar in other cells expressing those ion channels. So the next research question we aimed to determine was, is there a functional change in TRP ion channels on the cell surface of natural killer cells from ME-CFS? Now, determining a functional change is critical in terms of looking at calcium mobilization into a cell. So how did we actually test for TRPM3 function? For those people that have followed our journey, you would understand that this has been an extensive journey where 18 months has taken to actually complete the analysis of this methodology. And the methodology we used was patch clamp electrophysiology. Now you might ask why? Well, that is actually the universal gold standard for the identification of impaired ion channel function. So what is patch clamp? Patch clamp methodology, if you cast your eye over to the top left-hand side, you can see this diagram depicts cells. The cells in this instance are isolated natural killer cells. Now, if you cast your eye to the lower left-hand side here, this schematic is a cartoon of these cells isolated. What you see here is an isolated natural killer cell in cartoon form, and you see the patch or the electrode that has made a seal on the cell surface, and in this instance, the ion channel, TRPM3. Now, what does that actually look like? The TRPM3 ion channel is represented here, and subsequently it allows, upon stimulation through threats, as Professor Stones has outlined, the mobilization of calcium inside the cell. Now, what actually causes the function of TRPM3, as Professor Stone said, was a number of threats, but we can't mirror those threats in a test tube or in electrophysiology conditions. So we mirror that threat by adding an antagonist or an agonist. Now, in this instance, the agonist is actually known as pregnenolone sulfate. Here, this diagram here on the right-hand side depicts the agonist, pregnenolone sulfate. When pregnenolone sulfate is added into this electrophysiology methodology experiment, it initiates the activation of TRPM3, and as this diagram shows, allows then the mobilization of calcium inside the cell. Conversely, if we look at the other pharmacotherapeutic drug, onanetin is an antagonist, meaning it blocks TRPM3 function. So we use onanetin to block TRPM3 and therefore prevent calcium ion mobilization inside the cell. So with this method, we can actually see the ion channel opening and consequently also closing. So when we look at what we have reported in the literature, we have determined that we have significantly lower levels of calcium entry inside the cell. We have significantly lower levels of calcium stored inside the cell. We have significantly lower TRPM3 expression on the cell surface, as well as significant associations of TRP ion channels in ME-CFS patients compared to controls. So this functional test we undertook with patch clamp electrophysiology was the gold standard to determine if these ion channels were faulty. Now, we haven't actually done it in one particular ME-CFS cohort. We've actually repeated this experimental design in four separate cohorts of ME-CFS patients that were also age and sex matched to healthy controls. Now, this schematic here on the left-hand side is an example of one of the 4 studies we have reproduced and reported, TRPM3 dysfunction. What you see here on the y-axis is TRPM3 current after pregabalin sulfate, as outlined here, the agonist. And in the healthy control, you can see it responds in a normal fashion. That is, it allows calcium to enter into the cell. In contrast, when we add in the agonist, pregabalin sulfate, to ME-CFS patients' cells, we see there is a significant reduction in the opening or the function of TRPM3. Similarly, another example of these 4 studies is shown here on the right-hand side. TRPM3, again, current, is represented here on the y-axis with the subject populations, namely the healthy control and ME patients, on the x-axis. Again, when we stimulate pregabalin sulfate with the for TRPM3, we see the healthy controls respond in a normal fashion, and in contrast, ME-CFS patients do not respond. That is, their TRPM3 function under agonist activity is not functioning. Now, what does this look like in terms of real time? So this schematic here shows a healthy control where we add in the pregabalin sulfate represented here, and you can see the agonist opens the TRPM3 in the healthy control. Whereas when we add in the onanetin, you can see it tracks downwards. So it's antagonizing or blocking TRPM3 function and thereby reducing calcium entry inside the cell. Now, when you look at this schematic over here on the right-hand side, what you see is this is the normal baseline for ion channel function. And when you add in the pregas, that is the activator or agonist for TRPM3, you see you get this nice response passing through nearly the 0 axis. In contrast, when we look at ME-CFS patients, the same methodology of adding pregabalin sulfate and as an agonist and looking at the TRPM3 ion channel function, we see it is less responsive, and similarly with the antagonist onanetin, again, little response. Now, what we see in real time is represented over here on the right-hand side of this slide, where there is no arc for the pregas response. And this is typically what we have found for every ME-CFS patient for every study. Now, I'll just caution here and say, you might think, well, is it one reading we're doing for each of these patients for each of these studies? And the answer is no. These studies that we have reported for 4 separate cohorts with age and sex match controls, we have at least 6 readings per patient and per healthy control, all demonstrating the same response depicted on this slide. So what does that mean in terms of where are we heading with a test? What does a test mean? A test has to be valid. Okay, a test for a screening test or a diagnostic test, it has to have reproducibility. Now, when we apply the patch clamp methodology tests we have developed, as I have just said, 100% of all ME-CFS patients defined by the Canadian consensus criteria in all 4 studies had TRPM3 dysfunction, at least 6 readings per patient, per study. Importantly, this significant impairment in TRPM3 from isolated NK cells in ME-CFS patients is a clear marker compared to healthy controls. So now we have confidence, and we are reproducing this not through publication, but in our laboratory as we are speaking, actually. And we have now worked towards a laboratory developed test with the aim to develop NATA accreditation. So that is the next phase, the national phase for NATA accreditation for this actual test. Now, in addition to this, we have also a way of determining the responsiveness of TRPM3, and that is through using low-dose naltrexone or naltrexone, because they are 2 separate drugs in different dosages. And that means we can actually measure a patient's responsiveness to low-dose naltrexone and if it might be advantageous for them in a clinical setting. So if we look at this diagram here, in summary, the test we have developed is particularly relevant for TRPM3. We're showing it's got a significant reduction in calcium entry through dysfunctional TRPM3 receptor, and that is universal, we have reported, for ME-CFS patients. So where does that take us? If we've looked at and identified the pathology for ME-CFS, and we have great confidence now in our laboratory developed test, we're now targeting particular drugs that are for repurposing purposes for TRP ion channels, and in this instance, TRPM3. What we can see is we need a suitable treatment that must be obviously able to restore TRPM3 function, and it must be compatible and provide evidence through systematic review that it is a viable pharmacotherapeutic. And importantly, a pharmacotherapeutic universal treatment must have a biological plausible hypothesis and have proven data, reproducible. So where are we with this? We're actually, last year we published a paper looking at a particular receptor, and it's called the muoid opioid receptor, labelled here, and it acts as an inhibitor to TRPM3, outlined here. Interestingly, naltrexone inhibits the muoid opioid, allowing then for calcium entry into the cell. So when we added naltrexone into the in vitro situation of electrophysiology, we reported that we had restoration of TRPM3 as outlined on this slide. Patients that had provided their blood and were incubated overnight with naltrexone had restored TRPM3 function. And when we compared them to patients and healthy controls outlined on the right-hand side here, we see that they have restored back to normal function. So in this instance, we can see we have an impaired calcium TRP ion channel known as a channelopathy. And it's showing that we have abnormal movement of calcium ion channels into the cell. So where are we now? We now are focusing on this pathomechanism with looking at TRPM3 dysfunction across Australia with our collaborators, Professor Livia Houle at the University of Western Australia. We are continuing our MRI imaging, and we are now focusing on isotyping of TRPM3. At this time, I'd like to thank the team. It is a team effort, and importantly, the patients. The patients are why we work here. And also, if we haven't got participation and belief from the patients, then we can't make this research happen. Thank you.